everyone, welcome to the channel Question to Cure. This is the start of a medical research series where we explore large, complicated health science topics and break them down into small, bite-sized videos for any of us to learn to think like a researcher. Let's see what we have for today.
When you picture a research lab, you probably picture the movie version. Dramatic lighting, someone in a white coat leans into a microscope and gasps. I want to tell you what it's actually like in there because it took me a while to find out, and honestly, nobody shows you. It is slower than you think, it is far more social than you think, and there's an enormous amount of coffee. Start with the things in the room. First, there are pipettes. You will spend more hours holding one of these than doing anything else. They move tiny volumes of liquid from one tube to another thousands of times. And there are freezers that run at negative 80 degrees Celsius humming in the corner, packed with little labeled tubes. Each one of those tubes might be a year of somebody's life. When a freezer fails overnight, and they do, people cry. Then there's cells growing in incubators, microscopes, obviously, but also laptops, because a staggering fraction of modern technology is just code and statistics. And there are whiteboards, covered always in half erased arguments. Now, the part that nobody explains, who's actually in there? At the bottom, there are undergrads, learning the instruments, doing a lot of washing up, occasionally being handed something real. And then there's technicians and research assistants who keep the entire operation alive and are almost never mentioned in the paper's headline. There's PhD students, each one chained to a single question for something like 4 to 6 years. Their whole degree hangs on it working. There are also postdocs, people who already have a doctorate and are now doing the bulk of the hands-on science. Honestly, in most labs, the postdocs are the ones making the discoveries. And at the top, the principal investigator, the PI. The names on the door, on the papers, on the grants. Here's the irony that surprised me the most. The PI is usually the person who touches the equipment least. Their job is writing grant applications, asking for money over and over so that people below them can keep working. Now, the honest bit. Most of what you do does not work. The cells get contaminated, a reagent went bad, the freezer failed, you mislabeled 2 tubes back in March, and every result since has been meaningless, and you only realize that now. A normal productive month contains a great deal of failure and one thing that finally works. And this is why when people list the qualities of a scientist, persistence always shows up, and it always clicks like a cliche from a careers pamphlet. It isn't. Persistence isn't a virtue in this job. It's the job description itself. The experiment doesn't care how clever you are. And so much of research is simply waiting. Cells grow at the speed cells grow. You cannot rush an incubation. You set something running on a Friday afternoon, and on Monday morning, you find out whether the last month of your life was worth anything. The gaps aren't wasted, though. That's when the arguing starts happening. The lab meetings where someone presents their results and everyone else tries kindly to find the hole in them. That's peer review, and its earliest and most useful form. Your friends in a room refusing to let you fool yourself. So, slow, social, and mostly unglamorous. A lot of failure, a lot of waiting, a lot of coffee. And every single medicine you have ever taken came from a room that looks exactly like this one. That's not a disappointing ending. I think that's the point. Discovery isn't a lighting strike that finds a genius. It's a normal person in an ordinary room doing careful work on a Tuesday.
If you enjoyed this video, be sure to check out the rest of the series linked in the description below. Bye!